Good morning, everyone. Today I will talk about a custom meat project. It's a project that was created in Belgium between two Belgian universities, KU Leuven and Ghent. And it encompasses different topics. We actually studied 3 different cell types: myoblasts, bovine myoblasts, adipose-derived stem cells, and also embryonic stem cells. But today I will talk about specifically about myoblasts and their struggle to differentiate. So to date, in the scientific literature at least, myoblasts are the most used cell type for the development of cultured meat. So we did this systematic study where you can see that we found that, until 2023, publications that were about myoblasts specifically studying cultured meat were the most abundant. And then also more publications are emerging about MSCs and ESCs. And the reason of that is that it's a cell type that is easy to source. It lacks genetic modification, and we discussed that yesterday. Should we really stick to that? But we decided to, when this project was drafted 4 years ago, we decided to stick with that notion. And it has rather straightforward differentiation procedure. And yes, today I will talk about how they lose their differentiation capacity with time, but at least in the beginning, it's rather easy to induce myogenic differentiation into muscle. So I always like to refer to this slide as a reference point of what do we need in terms of cell quantities and at which point we will want to differentiate them and actually create muscle fibers. So based on what was published, first cultured burger was created from about 15 billion cells. And it takes about 18 doublings if you start from a biopsy where you isolated, let's say, 50,000 cells. And then if you want 1 kilogram of cell mass, you will need about 22 doublings. For 1 cow equivalent, we already go up to more than 30 doublings. And more than 60 doublings, theoretically, will be able to replace all cows in the world. But let's see if they can actually do that. So first, when we isolated cells from a Belgian blue breed, our estimate was actually pretty good, of 50,000 cells. But from fetal donors, we actually isolated more cells. And that was not unexpected, because fetus is still a developing organism and it has a lot of progenitor cells. And we isolated in the spread, the standard deviation is pretty high, but we were able to isolate hundreds of thousands of cells from a half-gram biopsy. From adult muscle tissue biopsy, we were able to isolate about 40,000, 50,000 cells. Then, when we actually checked their proliferation, and you can see here on each line represents an individual donor, we realized that yes, they divide rather quickly, and within 30 days, you're able to usually achieve 30 population doublings, and that's actually, so it's good news. And even so, they were able to surpass sometimes 60 doublings. And from adult cells, we had way more often cells that were able to surpass 60 doublings than from fetal cells. So in fact, it doesn't matter. You don't need to use fetal cells if you are targeting more proliferation. But let's look at how their doubling time changes over time. We found that in these adult cells that proliferated further than 60 doublings, we saw often an event where after high doubling time, when they were slowing down, almost reaching doubling time of 3 days, at which point usually cell culture stops dividing. These cells after that started dividing faster, and perhaps it is spontaneous immortalization. Perhaps there is mutation in some genes that gave rise to a population that divides faster, and that cell population continued dividing. But we also wanted to check what are those cells, if there's any major chromosomal aberrations in those cells, and there were none actually. So we performed this shallow genome sequencing to see if the number of chromosomes is more or less the same. And according to the reference cow genome, which was a different breed, so you can see, for example, a deletion in X chromosome. But overall, there is no major chromosomal aberrations. So those cells are rather genetically stable. I cannot pinpoint specific mutations, of course, because this is done per 100,000 base pairs. But overall, they were genetically stable, so which might help you with a further approval procedure. But actually, the topic of this conversation is, can they form muscle fibers? Because that is the main struggle with this cell type. And here, a minute of self-promotion from our lab. Our lab published pretty good software to check fusion index of the cells. There's been others, of course, published previously, but if you're still looking into a way how to quantify this in a rather quick and accurate manner, we published this based on existing AI-developed algorithm called DeepCell, which pretty accurately determines, separates nuclei and able to count those nuclei and able to calculate the proportion of cells within myotubes and outside myotubes. So it's an open source software, and you can check it if you would like to use it. So when we actually checked our cells, how well do they fuse, we quickly realized that fusion capacity or ability to form myotubes decreases quickly with time. And in adult myoblasts, it's very dramatic already between first passages and third, fourth passage. That decline is very steep, except for one donor. One donor also, Mark said yesterday that there was one cell line that also continued fusing. We also had that kind of cell line that continued fusing. But the rest of the adult donors could not fuse anymore after expansion. With fetal cells, we found that they maintain their, first of all, their differentiation capacity in the beginning is a bit higher, is about 55%, compared to adult, which is more around 40. Fetal cells also lose their fusion capacity at the moment where we want them to fuse. So actually, after expansion, they are not able to fuse well anymore, and they lose their fusion capacity about by 7% each 3 to 4 doublings. So we tried many things actually to optimize their culture conditions, but it's a bit shooting in the dark. We basically looked in the literature and saw what can improve their differentiation capacity, and we tested a bunch of conditions. We did not do all combinations of these conditions, but we did try a p38 inhibitor that was published before, and for this breed especially, it's important to use p38 inhibitor because Belgian blue breed lacks myostatin. Oh, it has a deletion in a myostatin gene, and which makes them differentiate prematurely. You don't want that to happen, so it helps a bit. Also, fetal cell source, as I said, was slightly better. Also, culturing them on laminin instead of gelatin improved slightly their differentiation capacity. And we even tried a different type of dissociation enzyme, which hopefully will be published soon. But none of them really dramatically improved that. Then we tried a combination of all conditions to see whether that will help us to achieve better fusion. And the best result, basically, if you boil it down to what matters for cultured meat, is that about 20%, you will basically get about 20% of myotubes in a cell mass anywhere between 200 grams and 200 kilograms. So not the best results, but instead of shooting in the dark like this, I'll go back to the previous slide and show what we did to find out the omic changes in those cells. So essentially, we took a selection of those cells and divided them in two groups. Cells with better fusion capacity and cells with worse fusion capacity, and compare their transcriptomes and proteomes. And today I'll talk a little bit about transcriptomic results. So we found that, first of all, most of the genes that were differentially expressed were downregulated. About 88% of genes were downregulated in the cells that cannot fuse anymore. When we try to make sense of that data, we use several bioinformatics tools to actually see what's going on. And the first thing that we found, of course, there was downregulation of myogenesis. Yeah, of course, we already saw it on the images. But what was important is that among the myogenic regulatory factors, myogenin was downregulated. And also myogenin's inductor MEF2C was also downregulated. What we found in literature is that MEF2C can be repressed by HEY1 activity, and HEY1 signaling pathway was actually upregulated in our myoblasts with low fusion index. So there might be something, there might be a way to actually repress HEY1 activity and see if that in turn leads to improved myogenesis. Second thing that we noticed was that there was a lot of genes associated with downregulation of DNA repair mechanisms. So in fact, when cells divide, they make a lot of there's often mistakes that is done in a DNA during replication procedure. And satellite cells especially, they're designed to be divided asymmetrically often, but we don't allow that. Basically, we put them in a medium containing a lot of mitogens and push them to proliferate further and further without chance to repair that. And perhaps a protocol which allows either slower divisions or periods of rest, periods of quiescence, would also help with myogenesis. And the last thing that I want to talk about is what we found is that there was upregulation of Rho GDI pathway and primarily driven by the upregulation of the LIMC2. It's a kinase. This kinase blocks cofilin. Cofilin actually depolymerizes actin, and actin depolymerization is very important for myoblast fusion. And that's maybe is what prevents cells from fusing. So actually blocking this kinase would be a rather easy, straightforward way of trying to improve myogenesis. There is other pathways which I will not have time to talk today about, but please come to me and we can discuss that further. So the conclusion is that yes, bovine myoblasts are they are capable of large-scale expansion. Unfortunately, they do lose their fusion capacity, and there are many genes that can be targeted to try to improve myogenesis in those cells. So the project has ended, and that's why this is my open call for everybody to just talk to me and see if we can continue, maybe write a grant together and see where this can go. And with this, I would like to thank you all and the Custom Meet team members and the universities that I worked at, my colleagues. So thank you very much.
Maybe you can stand here for the question. Thank you so much. Great talk. So we have time for some questions. Any questions? Yes?
Interesting talk. So you notice downregulation of HEY1, which is a downstream target of the notch pathway. So I was wondering if you're considering adding boosters of the notch pathway to see if you increase myogenesis.
Yeah, it can be definitely an approach. I myself am not specialist in the HEY1 pathway, but I did read that indeed it is, I'm not sure if notch activates it but then we actually want to repress it. So because HEY1 actually represses the activity of MEF2C, and MEF2C is an inductor of myogenin. So instead, in this case, I would inhibit notch to try to trigger this process. But again, maybe we need to do that in the periods of time. If notch is essential for proliferation, for example, and we want to, and as we saw, we had problems, if there is DNA damage associated with replication, perhaps we can induce quiescence in that manner, but yeah.
Yeah, no, it's interesting because notch in the mouse at least increases satellite cell proliferation. So
but that's the thing. If we can repress proliferation that way in a periods of times and that improves overall cell performance, maybe that's the way to go.
Thank you.
Any more questions? Yes, one in the front here. Oh yeah, was it? Yeah.
I'm curious if you have tried to do deeper genome sequencing, especially in that one clone that was mentioned that keeps fusing very well, sort of to figure out what specific mutations you could potentially, going back to the GMO topic, introduce in those lines that fail to differentiate just to make it go. Because I'm kind of wondering how much the information, very useful scientific information you found can be actually leveraged in protocols that will become more and more complex as you add additional factors, and also regulatory-wise, it may increase difficulties.
Yes. Yeah, of course, there are more advanced tools that we could use for sequencing. We could also, one thing that we wished to perform was like a full genome sequencing, also to see what kind of breaks do we get in which kind of genes. Unfortunately, we didn't do that also with financial constraints, but it's something that if we would continue working on it, we could definitely do.
Okay, one final question. Yeah, you see.
Thank you. Very useful information. I don't know if my question is needed because you still found such useful insights, but I was wondering how much the purity of the cell population, could it be like fibroblasts, overgrowth, that plays a role in this? Yeah,
indeed. We mostly checked CD56 marker that I did not really mention, but CD56 marker in muscle-derived cells are mostly myoblasts. Of course, it doesn't say which subpopulations of myoblasts it is, but because we wanted to study what is the problem in myoblast population, we in fact took cells of early passage, so passage 1 to 2, and then the later passage was passage 3 to 4, so not a big difference. But in that period of time, actually the population of CD56 positive cells was high. So it was more than 90%. So yes, it's a bulk RNA sequencing that can include other cell types, but because it's still a majority myoblast cell population, we thought that's where we can compare the transcriptomes. So, but yeah, after like around passage 6, also there is fibroadipogenic progenitors that start emerging. But the fusion index reduction was actually preceding that. So I think the overgrowth of fibroadipogenic progenitors is not the main driver of this reduced myogenesis.
Okay, then I say thank you again for this nice presentation.
Thank you.